test is subdivided into three. First is preparing the sample. Step 1. The blood is thrown by capillary action into the capillary tube. Step 2. Allow the tube to fill 3/4 full by capillary action, which are then sealed. Fill the second tube in the same manner. Wipe the outside of the filled capillary tube with a soft tissue if necessary. Second part is the centrifugation of the sample. For centrifugation, the sealed tube are placed in a radial groove of a special microhematocrit centrifuge. The sealed end of the tube are placed against the rubber gasket and the open end toward the center. Ballast the centrifuge by placing the tube opposite to each other. The lids of the centrifuge are secured and the tube are centrifuged for 3 to 5 minutes. And the last part is reading the microhematocrit. For reading, it uses special hematocrit reader. It also should be performed in duplicate on each specimen. Average of 2 is reported. Hemoglobin determination using acid hematine method. First thing to do is to assemble the materials and equipment that you will be using for the test and also prepare the sample. Second, fill or graduate the tube of the hemoglobinometer with the hydrochloric acid up to the 2 mark on the gram scale. Third, draw a blood into the pipette up to the 20 mark and expel the blood into the graduated tube. After thorough mixing, allow it to sit for 10 minutes. After 10 minutes, add the distilled water, drop by blood, mixing after each addition until the color of the solution matches the color of the standard. Take the reading corresponding to the height of the solution and report the result in gram per 100 ml of blood. Manual WBC count. First step to conduct this test is to prepare the materials, diluting fluid, and samples. Second is the dilution of blood. Draw blood up to the 0.5 mark in the Thoma WBC pipette. Holding the pipette almost vertical, place the tip into the diluting fluid and draw the diluting fluid into the pipette slowly until the mixture reaches the 11 mark. Next, place the pipette in a horizontal position and firmly hold the thumb over the opening in the tip of the pipette. Detach the aspirator from the other end of the pipette and mix the diluted red blood cell count using a mechanical shaker for 3 minutes or by hand for 1 minute. Second procedure is filling the counting chamber. Hold the pipette in a vertical position with the right index finger covering the top of the pipette. Discard the first 4 drops of the mixture onto a piece of tissue. Using the index finger to control the rate of flow, place the tip of the pipette on the edge of the ruled area of the counting chamber. Fill the opposite side of the counting chamber with a second white cell count dilution. Last part is the counting of white blood cells. For counting, place the hemocytometer on the stage of the microscope. Use the low-power objective to focus. The white blood cells should look like small dark or black dots. Scan the 4 large corner squares and count the cells and record. For calculation, follow the formula shown. White blood cell differential count. Differential blood count gives relative percentage of each type of white blood cell. It includes neutrophil, basophil, eosinophil, lymphocyte, and monocyte. Here are the pictures of the white blood cells under the microscope. Platelet estimate. To do this, read 10 fields and count every platelet found in one field. Next, get the average of the 10 field and multiply by 20,000. Here is the example of a platelet under the microscope. Automated CPC and platelet count using Sysmex. first step is to input the patient's information into the machine. Next, you would see a green light which indicates that the machine can aspirate the sample now. Just click the button. After, you would hear a sound which indicates that aspiration is done. After a few seconds or a minute, the results will be flashed on the screen. Now the result is out, record the result into the form. Prothrombin time using CL analyzer. First step is to add 200 UL of the reagent into the cuvette. Next is to incubate the reagent into the machine. After incubation, you can now do the testing. Put the cuvette into the reading cell chamber and add the plasma. After adding the plasma, the machine will run the test and the result will be flashed on the screen. Always remember to dispose the cuvette. ESR. In this test, you will be using a block top evacuated tube. First is to mix the sample thoroughly. Slowly insert the graduated tube into the evacuated tube. Make sure to do it slowly to avoid bubbles. Push the tube until the blood reaches the top. Place the tube in a flat surface and sit it for 1 hour without being disturbed. After 1 hour, you can now read the results.